Can you hear me, sir? Check this, sir.
Hello.
Sir, can you hear me?
Am I audible?
Yes, sir, you're audible.
Hello. Hello. Okay. Hello. Yeah.
Good evening, all of you. Am I audible? Am I audible?
Yes, ma'am.
Yes, ma'am.
Okay. Thank you. So today we have our resource person as Dr. Vinayaka Hegde, and I'll give a brief introduction about Dr. Hegde. And regarding his academic qualification, Dr. Hegde has done his post-graduation in agriculture as well as his PhD in plant pathology from IRI New Delhi. And saying a few words about his achievements, he has developed diagnostics for diseases of tropical tuber crops, and he has first-hand recorded the mixed occurrence of SPFMB and sweet potato leaf curl Georgia viruses in India based on sequencing of CP genes of the respective virus. And again, Dr. Hegde has developed serological and nucleic acid-based diagnostics for the detection of sweet potato feathery mottle virus as well as Dashin mosaic virus. And he and his team has developed an integrated disease management strategy for cassava mosaic virus, which involved the use of meristem-derived virus-free cassava and planting of barrier crops like maize or red gram, spraying of antiviral plant extracts like Behavior diffuser, etc. And Dr. Hegde has also developed a simple and efficient micropropagation protocol for greater yam and taro, indexing of potato for viruses. And he has indexed more than 500 tissue culture sweet potatoes for viruses by electron microscopy and serology-based detection of potato viruses. And now Dr. Hegde is the head of the division as well as the principal scientist of the Division of Division of Crop Improvement at ICAR CPCRI. And in his, he had bagged a number of awards, which includes IRA merit medals for outstanding research work during his PhD work, as well as UAS gold medal for highest OGPA during his PG days. So we are fortunate enough to have him on this session, and I welcome you, sir, for the session. I also welcome all the students. Sir, start the session, sir.
Okay. Thank you, Dr. Nima, for the introduction. So I will be speaking on coconut disease today, as mentioned earlier. Let me try to share my slides. Is it visible?
Yes, sir, it's visible, sir.
Okay. Okay. Good evening, everybody. I hope you're all students of agriculture background, BSc or MSc in agriculture. So probably you must have idea of the disease management in different crops and different ways how the different diseases are being managed. This will be on coconut. I will just share our experience as well as the work major diseases, so that in the classes where all the diseases are being dealt, it will be a very brief But here I would like to give you the major diseases, and we can have an interaction if you have more interest in the disease to know more about that, and how we are advocating the management of this diseases. I would like to tell in few minutes all the major diseases of coconut. 1 minute. The question is why we need to protect the farms, or plants from the diseases. You know, the global population is increasing, and increasing demand for food as well as horticulture produce. There is even for the coconut, you know, the tender coconut, lot of demand is there. Even coconut, there is a demand increasing as a health food. So we need to protect these foods and increase the production. So more than 1.2 billion people went hungry in 2009 as per the FAO. Of course, again, it is more now, recent reports. Global production must increase 50%, they told, by 2050. So we have to, one of the major issues in any food crop or any horticulture or any other crops, the major is pest and diseases are one of the major factors in reducing the disease yield. So you can see here the last 40 years, though we had chemical for the doubling the food production increase, still we are these pathogens or diseases, they claim about 10 to 16% of the global harvest. So overall around 6 to 12% we are losing in the post-harvest, even after the harvest. So in some of the terms, around $220 billion US dollar is lost due to diseases. This is the report in some of the international reviews. So this is the where we have to go for protection of this disease. So one of the ways is to know the disease and how they're spreading and how to stop those diseases and manage the disease, because we cannot control any diseases. Once it is there, it may be difficult to eliminate, but we should know how to learn to live with certain diseases and how to eliminate certain diseases and how to manage the diseases to get a sustainable yield. So coconut production, if you see, come to coconut, you know that more than 100 countries it is being grown. India is one of the largest producer and consumer of coconut. It provides livelihood security to millions of people in Asia as well as Pacific regions. You know, culture and traditional importance it has got. You know, every house in India, most of the places you may be knowing, before the house construction itself, there will be every house, there is 1 or 2 coconut trees will be there, mainly cultivated by small and marginal farmers, and increasing demand as health and functional food. There is a demand now with the awareness among the people to go for more and more health produce. So coconut being a health promotional food, it is being increased demand is there. So this coconut is also called as Kalpavriksha. You must be knowing, the tree of life. So every part of the coconut is useful to mankind. So it is in India, as I told, it is being grown. You must be knowing major the Kerala. The name itself is from derived from coconut, Kera growing area. So where it is called Kerala and Tamil Nadu, Karnataka, and Andhra Pradesh. These 4 states accounts for more than 90% of coconut produce growing areas in India. And it is also being grown in some of the West Bengal, Orissa, as well as, of course, northeastern states. So present productivity is 1 ton copra per hectare per year, and mainly used for coconut is for consumption, indigenous consumption. We are not able to produce much like other countries like Southeast Asian countries produce and export. But because we ourselves are consuming most of the coconut produced in India, so capacity 3 to 6 tons depending on input management. If you manage properly. So world area is this, as I told, 12.47 million hectare. And in India, you can see the productivity, average productivity is 9,000 nuts per hectare. So this is what in general production in Coconut Development Board, you can get the statistics, recent statistics all. So then I will come to my today's topic as why to protect coconut. So as a pathologist or as a plant protection person, you must be thinking why to protect coconut. There is no protection for any wild plants, and it is growing and we are harvesting. Why we should go for protecting the coconut? So you know that it is the livelihood and loss due to natural calamities. We know that many years during cyclones, last many times, even in Gaja cyclone in Tamil Nadu or in Odisha, lot of damage due to for coconut. And this flood in coconut in Kerala also, there will be a lot of diseases you can see, as well as sustaining production and productivity is essential. We should protect our coconut being a perennial crop. So second issue is why with safe chemicals and biocontrol agents. As you know, there are a lot of issues over the years. We have recommending in many food crops and this one different chemicals and different pesticides to increase the production. But there are certain issues flared up. You must be knowing about the endosulfan issue or any other production nowadays, the spurious chemicals being marketed, which are the that lead to not being used properly, misuse of the pesticides, which led to a lot of problems in hazards. So pesticide residues in the foodstuff, environmental pollution, imbalanced ecological equilibrium, resurgence of minor pests and pathogens. So we need to balance our food control as well as manage. At the same time, we should be able to manage our environment, food hazards, and other ecological balance we should manage. Otherwise, even if we kill certain pests or diseases, then others will, other minor pests will become major problem. So we should see that there should not be any equilibrium should be disturbed too much. So we should be minimal. This one, we should be able to manage the disease to get a sustainable and good yield from our produce. This is what our, with this in objective, I think we have to go for managing and understanding the diseases. So this is the one loss in coconut. You can say that is a biotic loss, pests and diseases and wild animals. These are some of the pests. So you can see the red palm weevil, Dinoceras beetle, and different diseases like bud rot. These are the major diseases and pests here in coconut. And abiotic stress, like different deficiencies of nutrient or moisture stress and toxicity of certain elements, all cause different types of problems. And natural calamities like cyclones, floods, drought. You can see a lot of damage during the last cyclone, Gaja cyclone in Tamil Nadu, or even in many other places, like good trees. Farmers have lost several thousands of trees have been uprooted. So this is a major issue. So coming to the pathogens or a disease per se, as what are the major issues involved, we can see that there are the major, even in I have put is arecanut also, because most of you know that arecanut is also increasing, and most of the coconut farmers in the Kerala, Tamil Nadu, or Andhra, or Karnataka, now they are shifting towards arecanut also because of the price. So, and most of the pathogen problems are also similar. So I just put arecanut also here. These are the three pathogens of major threat to coconut. As I have, there are many pathogens, more than 120 pathogens are reported on coconut. But globally, if you see, there are Phytophthora, Phytoplasma, and Ganoderma. Ganoderma is that these are the three pathogens which is globally very important and major problems. And recent years in our place or in India as such, we are seeing there's another two pathogens. It's the Colletotrichum species, Colletotrichum, and Lasiodiplodia species. Earlier, it used to be called as Wateriodiplodia. Now it is called as Lasiodiplodia. So these two pathogens are creating problems as emerging diseases come as case of coconut, lot of leaf blight of coconut. It is major issues even in Tamil Nadu, like Pollachi, where the coconut major hub of coconut area. There also the major issue of leaf blight caused by this fungus, as well as in arecanut in recent years, it has come Colletotrichum blight, which causes leaf blight like this with yellow halo, and suddenly whole leaves are becoming blighted. So these are the two pathogens, both in coconut and arecanut, are emerging with the climate, probably change in the climate over the years, as well as the pathogen. It was not a major pathogen earlier, these two pathogens, though it was reported. Now, recent years, again, they are emerged as the major diseases. So coming to the major diseases, as I told coconut, there are 3, 4 or 5 major diseases, though there are more than 120 pathogens reported on coconut. So we can see that here bud rot caused by Phytophthora palmivora is a major problem, is a lethal disease, which cause about 3 to 5% coconut trees every year in endemic areas, especially in the heavy rainfall areas, in the hilly areas like Kutiadi or Calicut area, as well as even in Mangalore, Karkala and other areas. We are seeing that around 3 to 5% coconut trees are being killed due to this disease. So this is one of the major issues. Then another disease is basal stem rot. This is also caused by Ganoderma lucidum, another fungus, the Basidiomycetes fungus. Important disease aggravated due to, this is not, though not a very aggressive pathogen, or this is a very not a like a Phytophthora, but it can, once infected, it is very slowly kills the palms, and continuous drought periods. When we neglect, prevalent in all major coconut areas, this will aggravate the situation, and the palms are being dead, killed. So then another major issue you must have read in our BSc or this one, that is the coconut root wilt disease. It is another major disease. I will explain further. It is caused by Phytoplasma like organisms. I hope you all know what is Phytoplasma. Phytoplasma are bacteria like organisms, but devoid of cell wall. Cell wall will not be there. They are, that's why almost like bacteria, but without cell wall. So they are not susceptible to many of these bacterial antibiotics. They can not sensitive. So we cannot kill these Phytoplasma with antibiotics. So these are the debilitating disease. This is systemic and causes the debilitating disease. In case of root wilt, it is mostly prevalent in all the 8 districts of Karnataka and that Kerala and southern districts. More than 130 years it is present and slowly spreading to major coconut areas in Tamil Nadu also. So this is a major concern. Then leaf blight. This is, as I told, Lasidiopteria theobrami. This is another disease emerging. And stem bleeding. This is another disease where caused by the fungus again. This is not a major disease, but it is prevalent in most of the places. If we neglected gardens, suddenly it will increase, because the farmers initial stage may not take much care, because nothing will happen with this one. So, but over the years, it may occupy, then it may be difficult to control, and the palms will be succumbs to the disease. So coming to Phytophthora disease of coconut, as I told, this is the Phytophthora palmivora. Palmivora is, because of palm itself, it was isolated, and so it was referred as palmivora. So bud rot is the major issue. So bud rot means only the bud, the initial bud, base of the bud, it is infected. Fatal disease, it affects palms of all ages, but young palms in low-lying and moist situations are more susceptible, especially in the very humid, tropical, humid areas, because most of these are grown in coconut, highly humid areas. This bud rot will be the major issue. Bud rot disease is usually sporadic, so but sometimes appear in epidemic forms. Usually, we'll see here and there 1 or 2 palms like this. But what happens in certain conditions? This will increase suddenly because of the favorable condition and the source of inoculum in the garden. Like this type of plants, if you leave as such in the garden, the Phytophthora can survive in this crown region. And when the rain starts, from this portion to it will through wind blown rains, it will go to the next trees, and like that it will go on spreading from tree to tree. So this is one of the major issue. And the same Phytophthora can cause the dropping of nuts. You can see the infection on the nut like black spots, and this will affect the nut and it get loose and whole nuts are being fallen. This is the same even in other arecanut also. You must have heard the fruit rot or Coleroga or Mahali is a problem. Here also the fruits are infected and fallen, but the compared to arecanut, here the fruit infection is comparatively less, though it is bud rot incidence itself is more than the fruit rot incidence. Fruit rot also, certain cases suddenly it will be noticed, but the regular monitoring is very important because fruit dropping is due to this Phytophthora during rainy season is common. So we have to take prophylactic action for that. So withering of spindle, what is the symptom I have explained here? This is the first symptom of the disease. This is also already dried. Now only we can able to see. Before it will be just a drooping type will be there. So then that will be yellowing and this will be dried. So all other leaves will be green. So it is very difficult to detect at early stage. So when this type of situation appears, by the time if you remove this, it will pull off. It will come out of the meristem, that bud region will come out. And you can see here only the green leaves will be left and the portion will be rotten. So over the few months, it will become like this in the next stage. And this whole palms will be killed. This is the symptoms of the disease. You must have seen in any of the coconut garden, many places, it will be there without crown. That is due to this fungus infection. So this is the nature where you can see the endemic areas, how the palms without the bud, that this will be there. This is mainly due to the bud rot pathogen, Phytophthora infection. So this is the Phytophthora palmivora is the major pathogen which has been isolated and has been proved from the long back. Dr. Butler itself first time in 1904 itself isolated and established that it is caused by Phytophthora palmivora. And this is the sporangia type where you can see the papillate sporangia will be there. So this is the major pathogen involved, though some of the other pathogens are being isolated. However, we are not getting commonly, but the common pathogen for bud rot is Phytophthora palmivora. Then epidemiology, if you see that how it is a source of inoculum, rainwater, soil, fallen nuts remaining in the garden soil, common slug, and other susceptible hosts present in the garden are found to be the major source of inoculum for Phytophthora disease of coconut. See, Phytophthora can survive in the coconut or arecanut-based cropping system, which is very favorable because the moisture as well as the humidity within the plantations are there, and Phytophthora can survive throughout the year. With otherwise, any intercrops grown, so it will be growing there in the soil in the dormant condition. When the rain comes, it will be infecting. So this is the rainy season. This is the major problem. This is where you can see that these are the acts as a source of inoculum, fallen nuts. These are all infected with Phytophthora. So this itself will be there. It will act as a during rainy season, and the inoculum will be carried out. Then the question comes, why the bud is being affected, though it is in the ground level. The problem, these are some of the, you must have seen this in the coconut or arecanut or any area, which is called a common slug. So this used to be, we checked that this can feed on this Phytophthora and it get attracted, and Phytophthora is being spread from this also. This is also one of the carrier for Phytophthora disease in coconut. So what we have seen, you can see here the bud rot affect Phytophthora. So it is feeding on Phytophthora cultures also. We have put this, and we can see that it can go for feeding on this Phytophthora cultures. So this is also one of the issues in spreading the Phytophthora within the high rainfall coconut areas. Main reasons for higher bud rot incidence is prophylactic measures not taken up regularly. As you know that though we are telling easily that you should spray with prophylactic fungicide because Phytophthora will come, but what happens is it is not, the incidence is sporadic, and unless the disease comes, people or farmers are not ready to take up initiative for prophylactic action. So and the climbers for timely availability. So somehow it is not being taken up properly, and the reasons during favorable conditions, suddenly the disease incidence increases. Climatic changes and erratic rainfall. As you know, nowadays the rainfall is some years suddenly, certain months there will be severe rainfall and changing climate. So this is another reason nowadays, and non-adoption of scientific cultivation practices. Though in case of coconut, whatever the recommended fertilizers or this one, people are not adopting the cultivation practices. There is imbalance in nutrition as well, especially in the potash. Most of the gardens, you can see there is a potash deficiency, which is also one of the reasons for pest and disease attack. Potash deficiency leads to the susceptibility in the host. So that's the major issues. Then improper, inadequate use of fungicides. Even some farmers are using, many places the improper way or inadequate it was being used, that leads to higher incidence. So this is the integrated management strategy evolved for bud rot. Remove all the disease advanced and dead palms, destroy the infected crown region. Then crown cleaning, we should clean before the rainy season starts. We should clean the crown earlier, but it is easy to be said, but nowadays because of climbers maybe difficult, as well as the price situation. Coconut, sometimes there will be very low price farmers will be getting, so they are not going to enter into this type of work now. Field hygiene, as I told, the Phytophthora, this is one of the issues. Wherever we leave infected palms or the infected nuts on the palms, the inoculum load will be more. Adopt management practices recommended for rhinoceros attack. As I told, rhinoceros is another beetle which causes the damage to the spindle region. So where the rainy season, Phytophthora may come. So that is also, we have to see that it should be protected from the rhinoceros beetle attack. Then apply 1% Bordeaux mixture to the crown or crown area, because this is the place where Phytophthora affects. So as a prophylactic way, we should go for 1% Bordeaux mixture application, or Trichoderma coirpith cake. We have, I will explain this. This is the Trichoderma formulation developed at CPCRI. You can place inside the innermost leaf axil, which will help to reduce the Phytophthora growth. So pouring potassium phosphonate also was there, coming earlier into the solution. It is not recommended as such, but it is seen earlier that can also will help in protecting. So repeat the prophylactic fungicide or biocontrol treatments every 2 months interval in the endemic area, especially in the high rainfall endemic areas. Unless we are not prophylactic actions are taken every 2 months, we will not able to keep the palms free from bud rot. Observe all palms at 15 days intervals, because monitoring is more important. Every now and then they were to monitor once in so identify bud rot incidents, if any, in the initial stage, because if you are not able to identify the bud rot in the initial stage, then the difficult to cure the palms, and it will be killed. So continuous prophylactic fungicide treatment bi-monthly till the end of December. Actually post monsoon also, sometimes we are seeing that mostly in the December also the bud rot emergence will be there. Observance of all palms at 15 days intervals, and integrated nutrient management. As I told, that is another important of any plant disease management. We have to go for recommended dose of fertilizers, as well as other application has to be made. Bud rot is a curative treatment. As I told, all the palms in the garden once in 15 days during rainy season. Identify bud rot incidence in the initial stage. Remove infected tissues completely in the early stage of disease. Immediately after removing infected tissue, apply 10% Bordeaux paste. Cover the wound with polythene sheet, prevent entry of rainwater, and destroy the infected tissues removed by burning and deeply burying the soil. Follow prophylactic treatment with 1% Bordeaux mixture or potassium phosphonate. So this is the where I told you trichoderma talc formulations, and well as the trichoderma coir pith cake formulation. The trichoderma is embedded in the coir pith, the waste of the coconut product. So these type of cakes have been made. This can be kept in the room temperature for up to even 1 year. And once the rain moisture comes to this, you can see the trichoderma growth, this green growth. So this can be used. These type of things can be used as a biocontrol approach also to manage the disease of the bud rot. So this is the bud rot. This is about main disease about bud rot. So the question is how to do it. Why can't we go for any other approach? That I will tell at the end. Another major disease is stem bleeding. Stem bleeding is the another major disease which is prevalent in most of the coconut growing areas. This you can see here, the bleeding type of, there is a dark brown patches at the basal portion of the trunk, and exudation of dark reddish brown liquid from the trunk is the initial symptom. This is the symptom where you can say black encrustations will be there. So this is the type of symptoms if you must have seen many places in the coconut gardens, and we just chop off this portion. You can see this type of blackening of the interior inside also will be there. Gradually the trunk tapers towards the apex and crown size is reduced. So ultimately, if you neglect, if this disease is neglected, so it will lead to the reduction in size of crown and nuts production will be reduced. So overall, the palms will become unproductive. So stem bleeding disease is caused by the fungus Teleopsis paradoxa. So this is a soil-borne pathogen and enters the trunk through wounds and growth cracks. So these are the pathogens from one. So this is the managing this disease. Though it is not a very serious, which can, if you manage it, we can able to control. And though it's prevalent, we can get the good yield by managing the disease. This is the apply 5 kg neem cake fortified with 50 gram of Trichoderma hergianum talc formulation per palm at quarterly intervals. Irrigate the palms once in a week and mulch the palm basin with dried fallen coconut leaves. Smear the Trichoderma talc powder paste prepared by adding 50 gram of Trichoderma talc powder to 25 ml of water on the bleeding patches of the stem. That also can be made directly instead of applying. Where this is I am telling for where the Trichoderma they want to go for biocontrol. If there is some issues we are getting Trichoderma or the no much humidity where Trichoderma are not able to survive, they can have to go for chemical fungicidal treatment. With that, for this removal of affected portion with a chisel and smear the wound with fungicides like hexaconazole. Though carbendazole was there earlier, now there is certain issues you must be knowing that Government of India has listed certain 27 fungicides to be banned in the list, not yet banned completely, but it is there in the list. But alternatively, this is one of the good fungicide, that hexaconazole at 105 ml in 100 ml or 2 to 5 ml. In the severe cases, you can go for more. So that can be used for root feeding as well as applying onto the wounds. In cases of bleeding, when the bleeding patches extends towards crown, go for root feeding, because we cannot just smear it in all the places. If it is extended towards the crown, then you can go for quarterly root feeding with hexaconazole so that it can be managed. So this is the way in which the roots we have to find out, this type of feeding roots, and this where we have to fungicidal solution we have to root. So within 24 hours it will be absorbed, and this can be used for this will help in controlling the disease. So another major disease, as I told, is the Ganoderma wilt or Tanjavur wilt or basal stem rot. This is called as basal stem rot disease or Tanjavur wilt, because it was first reported from Tanjavur areas in coconut. So it is called Tanjavur wilt also. So this is caused by Ganoderma lucidum. So you can see here, this is a soil-borne pathogen. What exactly happens is it damages the root from the basal portions, whereas the symptoms on the top will be the leaves will become yellow and droop, and skirting of leaves like this will be seen in the severe cases. So farmers will start notice only when this happens, it's yellowing and drooping, severe drooping happens of the leaves. Though, and in the early stage, what by the time there will be the blackening or bleeding patches in the base of the stem. Sometimes it will be below soil. So farmers will not able to notice also from the below soil, and all the roots are getting damaged or rotting of the roots will be there. So then these symptoms, root and uptake of the nutrients affected, and the roots leaves will become yellow and droop like this. So the fungus also produce the what we call fruiting bodies, what we call in anabe or mushroom type growth will be developed at the later stage. You can see here already the palms are dead, and it is producing the this mushroom like fruiting bodies. So Ganoderma wilt or Tanjavur wilt, this is the symptoms we have given in a, this one. I will not go in detail. This you can get it in any of our books or even in our website. So this is the way in which it comes. So within the month of about 50 to 60, 54 months, it will be plants will be died once the infection starts if we are not taking any control measures. This is the way in which you are seeing. Even this is all in sea coconut gardens, where the affected portion in the on the basal portion. The basal portion, in case of stem bleeding, the infection and it will be reddish brown oozing, which will be starting from any point in the tree. Whereas in case of basal stem rods, this bleeding will be darker, and this will be from the mainly from the basal portion of the trunk. So actually what happens is the infection starts in the root and basal portion. So this basal portion of the stem starts getting damaged. So then whole leaves will become yellowing, and it will start drooping like this. So this is the typical symptoms in case of Ganoderma wilt. So it is called by Ganoderma lucidum mainly. There are another species also, Aplanatum species has been reported. Of course, there are a lot of work needs to be done, especially in the palms, whether it is entirely different from the other palms, which is like in oil palm or in the arecanut, whether it is the same one, though it is much studies have not available as of now in India, though it is the molecular works are being done now and then, but still not very clear whether they are the one and the same or it is different one. So that type of work is still going on. But as of now, the fungus involved is known as Ganoderma lucidum, based on the morphological has been identified, and the pathogenicity has been established also. Like epidemiology, if you say that it is a soil-borne pathogen, as I told, it's the coastal areas of coconut, then lack of soil moisture during summer months, water logging in rainy seasons, presence of old infections in the gardens. So it is the major, the soil-borne pathogen. Age of the palms of the age group of 10 to 30 years are more susceptible, 43% than the young trees. So more the years, the aged one are having more susceptible. Weather factors, disease incidence were more between March and August, and bleeding patches positively correlated with the mean maximum temperature. So temperature, the soil temperature, higher temperature, the disease incidence was also more. Time taken from initial appearance of the bleeding patches to the death of the palm is 6 to 54 months, and average of 24 months it will take complete death of the palms. In the middle state or stages, infestation of the stem at the bleeding patches by scotylid beetle. So Xyloborus performance and Weevil, Dioclandra sigmatis also happens, because this will damage the trunk portion of the bark in the trunk. So these beetles attack there, and it will become more become more severe, the disease. The insects accelerate the death of the palm. Once the insect attack comes, these insects, that will accelerate. So integrated management for this disease developed and advocated. So removal of dead palms and palms in the advanced stage of the disease, and destruction of the bole and root bits of these palms. Second, isolation of diseased palms. If one palm also noticed in that, we have to isolate by healthy palms by digging isolation of trenches of 1 meter deep and 30 centimeter wide, because the root, one of the major notice was the root contact of the disease to healthy palms is the one of the reason for spread of the disease. So that to isolate the disease with this one is one of the important issue. Regular basin irrigation during summer months is another, this one recommended. Stem bleeding disease, whether stem rot can also be effectively managed by soil application of neem cake enriched with 50 gram of trichoderma formulation. So this they have to regularly apply, and wherever the incidence is there, we can go for root feeding with hexaconazole or soil drenching, as I mentioned earlier for this also. And another observations made and in the earlier scientists also, that raising of banana as an intercrop wherever irrigation is possible will help to reducing the Ganoderma wilt disease. Wherever there is no intercrops and more incidence of Ganoderma wilt is noticed. So the banana somehow will be able to deter the pathogen probably. So that is known that banana intercrop is able to help to reduce the disease to a certain extent. Another major issues are immature or fruit rot. This may be many farmers may be asking if you also, or you must have also noticed, they are telling many of the nuts are falling, or this immature nut, small buttons are falling from the coconut, though good flowering is there and it's coming. So there are a lot of causes for considerable and direct loss. This is so because they are able to see that dropping of nuts. So they feel that there is a direct loss also. So this may be due to several reasons, this dropping of nuts. One is there may be genetic causes, soil factors, drought or waterlogging, nutrition deficiency, poor pollination, plant growth regulators, insects, fungal infection. So this may be due to any of these reasons, different ways it may affect. So we have to see the causes for this one, how it is by seeing the nut. If there is any fungal infection, we can adopt the management. Tender nuts of different ages, buttons before after fertilization, Eriophyid mite infested nuts, mature nuts, stored nuts, all this fungal infection will happen. So rotting and abnormal shedding will happen due to this fungal infection also. Immature nut fall, you can see here, this type of cracks also will come. One of these reasons for this cracking is known to be deficiency of potash and boron. This may lead to this type of cracking. And of course, fungal infection is also there. We can see the fungal infection must have led to the dropping of the nuts infection. This also. So that's you can see here, most of the nuts are infected with the fungus also. So even the fungus can enter inside and cause the rotting of the stem. This is see how the infected nuts are falling here, and this is one of the inside nut. So this is also how the rotting of the stored nuts happens. So one of the earlier recommendations was carbendazim. Of course, carbendazim nowadays, though it is under stress, we can go for hexaconazole also, 0.1% spraying. Otherwise, carbendazim plus mancozeb, the combi products are coming, that also will be useful. So garlic bulb extract, as well as that have also been tried earlier. So maximum of 2 sprays in 30 days interval will help to reduce the disease incidence. Wherever the diseases are there only they can go for spray. Then another emerging disease, as I told, this is the leaf blight caused by Lasiodiplodia. See, you can see here complete blighting of the whole leaf. So this is also seasonal and certain places. Sorry. So leaf blight can be managed with root feeding or with fungicides like hexaconazole at quarterly interval. We can manage the disease because it is difficult to spray for this pathogen, as well as it is airborne. So it may cause to, as a mass, they have to take up the control measures. It has been recommended to go for root feeding or for fungicides as well as some biocontrol agents nowadays has also been available, like Pseudomonas fluorescens as well as Bacillus. So that can also be tried toward the management of this disease. So these are the fungal disease. Another major issues is the phytoplasma disease in case of palms, especially coconut as well as arecanut. Coconut root wilt, as you know, it is caused by phytoplasma disease. So in world, phytoplasma disease are major in Caribbean region as called as lethal yellowing, whereas in other places also lethal wilt like disease are known. In India, we are not getting that type of lethal disease, but it is some sort of debilitating disease which was known to be caused by phytoplasma. Earlier, root wilt and Thatipaka disease were reported, but now we are not getting any Thatipaka disease is not there. Only in Godavari in Andhra Pradesh area it was there, but now recent years there is no such symptoms are seen or no such palms are available. Whereas root wilt is still a major issue in most of these areas. This is the lethal yellowing, which is not a problem in India, whereas in most of the Caribbean area, how it is happening completely, the palms are being killed within 3 to 4 months. They become yellowing and whole palms are being killed. This is, however, root wilt. It is not like this in case of India, where we are getting, you can see here, we are writing the wilt in the bracket. This was done because it has a long history of root wilt as a disease, has got a long history. This is the flaccidity, and earlier into 1882 itself it was reported, and a lot of scientists working on pathologists and the soil scientists thought that it is due to soil factors, and others pathologists thought that it is some initially as a fungus because it causes root rot. So, but finally during 1980s only, we could able to establish that this disease is caused by phytoplasma by observing the phytoplasma like bodies inside the infected tissues and proving the later pathogenicity also. So these are the symptoms which flaccidity of the leaves, like a wilting. So that's why it is also called a wilt. The leaves will become placid and rib-like like this, and this is the yellowing will happen and necrosis will later develop. So this is the way. So that's why it is called as wilting also. So root wilt means root has got lot of damage, the rotting will be there. So initially they felt that it is a root rot. That's why the root rot as well as wilt symptoms both will be there. So then later they thought that it's a root wilt disease like that it has come up. So yellowing and marginal. So this is the even inflorescence, there will be necrosis. So coming to the, even in the certain areas, we are seeing only the yellowing symptoms will be more prevalent in Tamil Nadu area, where we can see the root wilt affected in the adjoining districts of this one, Kerala, Kambam in Tamil Nadu. These are the root wilt symptoms. This is the major symptom is this type of flaccidity, marginal necrosis and yellowing, leaf tip breaking. These are the major symptoms where root wilt symptoms. And root wilt disease affected palms, more than 60% of these palms are affected by leaf rot also, because this is the leaf rot is caused by fungus. So once the leaf rot affected, this looks like this. This is only root wilt affected. This is already phytoplasma, this flaccidity is there. You can see here slightly bending of leaves and necrosis, whereas no leaf rots. Whereas when it is affected by leaf rot causing fungus, the leaf will become like this. You can see the severity. So this is the leaf rot is caused by the fungus. If there are different fungus like Exorahilum and Colatotrichum, so 2 to 3 fungus have been known to be causing this type of symptoms in the root wilt affected palms. So extensive rotting of the roots with progress of the disease, shedding of immature nuts, thickness 6. These are all the different symptoms. So oil content is very much reduced. So overall, the whole palms will become unproductive over the years. And this is what is major issues in major root wilt prevailing areas. This is the mid-whirl yellowing, and this is the inflorescence necrosis. This is the phytoplasmal bodies which have been identified through electron microscopy, and this we can see here involvement over the years. Then it has been ruled out other causing, and it is known to be caused by Phytoplasma. Initial years, they were using light microscopy and staining to find out the Phytoplasma presence. Later, with the chemotherapy, whether it has been known with the remission of symptoms, whereas later, when the PCR and molecular era, PCR has also been attempted to find out, and it has also been reported that it is caused by Phytoplasma belonging to the group 11th B. So this has also been identified and detected. However, still some issues with the regular standardizing these techniques, molecular techniques for detecting early detection of the disease, and early and accurate detection is using this molecular techniques is still being developed. There are certain issues for, especially for root wilt, though it is possible for other diseases, phytoplasmal disease. In case of root wilt and yellow leaf disease in arecanut, we are still struggling to standardize this techniques, though we can able to get certain times, but to use it routinely, this are not able to develop that type of kits. So efforts are on to go for a early detection kit for this disease also. Then managing, as you know, most of the phytoplasmal disease in the annual crops means we can just remove the palms and remove the affected plant. But as in case of plantation crops like coconut, it is not easy to remove the infected from easily. Nobody will be able to remove as soon as you get the symptoms, because or if the symptoms are 1 or 2 palms, also difficult to remove. Farmers will not able to remove immediately also. So that is the issues. So then how to manage it, since it is not killing the palms immediately, it can be managed to a certain extent by taking all the integrated approach. There is no direct control for this Phytoplasma. However, this is we can manage the disease by different approach like nutrient and fertilizer application properly, basin management. So this will overall reducing the other infection also, because once the Phytoplasma infected, what we have seen is if we neglect this type of tree which is infected, they will become susceptible to many other insects as well as this pest. So this management of this will take little more of efforts have to be taken, intensive efforts, then we can able to sustain the yield. So that has been demonstrated and had been we can able to sustain the yield. So this has been in different larger area. If we manage the disease, we can get the sustainable yield. Otherwise, if we are not managing, it is difficult to get a economic yield. That's what it has been demonstrating at different places. And CPCI, fortunately, in case of this one, some of the field tolerant and dwarfs like Kalparaksha and variety has been identified, and Kalpasankara, one hybrid, and Kalpashri. These three are known to be, this is a hybrid of, whereas these two are the varieties, Kalpashri and Kalparaksha. These are CGD and this green dwarf varieties, which has been selected and named as Kalparaksha and Kalpashri varieties. These are have got the have been multiplied and used under the endemic areas where root wilt is a major problem. So this is being replaced with these varieties. So control of leaf rot is a major issue. Wherever the root wilt is there, if you control the leaf rot, as I told, that is the one which causes the by the fungus. So that leaf rot, if you manage, so the severity will also reduce, and we can get the good sustainable yield. So we have to go for leaf rot can be easily managed, and management has been recommended for leaf rot also with the biocontrol agents as well as with the fungicides. We can manage the disease. Disease management, removal of disease affected in the mildly affected areas. We can recommend for removal of the immediately as soon as the symptoms comes. Whereas in the endemic area where already most of the trees are infected, in such case continuous monitoring. This is in the initial stage where there is no diseases are there in that area. If you find that type of palms, that can be removed. Whereas in the other area, we have to manage the disease with, as I told, different practices. Acclimation, these are the different ways we can get in our website also. Details of, I'm not explaining this. There is no direct control for any of the phytoplasma or viral disease unless we go for any molecular approach where we can go for that type of works are initiated, which may take time, especially in the crops like coconut. You know that how the we can go for producing the molecular approach to produce disease-resistant varieties of these trees. So as of now, these are the this one has been recommended. So major issues in case of this is spraying, as you told, in fungal disease like this, crop protection operations, we need either problem in the particular, in the recent is climbing and application of the this one. So in this NIT, I have not given here. Last few years, we have initiated use of artificial intelligence and drone-based application of crop protection measures for the for coconut, which may take few years, another few years. Probably even the farmers have also initiated use of drone for pest and disease management in case of coconut. Probably that may have to come using artificial intelligence, like detection at the early stage using the mobile or digital based detection of the disease and pest. So that type of work is being started, but it may take few more years to come to do that. These are some of the diseases I have written which are quarantine significance. As you know that these are not present in India, but this may come. So we have to be prepared for that. But of course, as per the disease is concerned, compared to these are long listing, this has not yet come to India, this, whereas certain pests in case of coconut like Eriophid mite or as of now the whitefly have come from different places. Now and then we are getting the pests introduced to India compared to diseases. So these are some of the issues. I have taken time for 1 hour and to explain a little bit. So if you want any questions, you can. Thank you. Thank you very much.
Thank you, sir.
Okay.
Session is open for discussion. Any questions?
Good evening, sir.
Yeah, good evening.
Hello.
Hello.
Sir, am I audible?
Yeah,
Yeah.
audible.
Yes, can you explain once again about molecular identification of depression disease you have told?
Actually, see, molecular means you have to go for either polymerase chain reaction assay for identification of the pathogen. So especially in case of Phytophthora, Ganoderma, all, once you get the isolation to confirm the identity of the isolated pathogen, we will go for PCR based detection. So that PCR based detection, we will go for a specific primer and amplification and sequencing. We are getting it done, and we will see that whether it is really the particular species. For example, as I told now, especially Colatotrichum species, lot of leaf blight species, as well as even in Acidiplodia, it is coming up. So we are trying to go for identification using the PCR-based marker. That is a must nowadays. Most of the journals or publications, they insist for PCR-based identification. That is one aspect. Second is in case of Phytoplasma also, the 16S RNA groups are available, that farmers, that Phytoplasma groups are there. There are several groups of Phytoplasma, over 35 different groups. So we are also trying at different ways of detecting this. Earlier, we have used different primers, which has already been recommended for Phytoplasma detection, universal primers. Then it was difficult to detect. Then we went for nested primers also. We could able to detect it. But the problem is, again, certain places, this Phytoplasma concentration is not uniform or it is not systemic. At a certain times it will be in the root, concentration will be more, sometimes in the bud. So that will be some issues will be there. We were checking at different stages at different time to detect this phytoplasma. So for that, we need to standardize still more standardization. Why we are not able to detect at a certain time and a certain place, though we are getting, sometimes we get the amplification, and when we give sequence, we will get the phytoplasma. But the same tree, if you take again, or the same at different place, the different stages, we were not able to get the phytoplasma again. So in that case, we were not able to use this technique to certify that seedlings are free from phytoplasma. So that type of standardization is required. That's what I mentioned. So other pathogens also we are using this. Regarding the molecular techniques for the management, as you told that nowadays different types of this double stranded RNA are being used. So that type of technique, or using transgenics, or using double stranded this siRNA techniques, we have to approach. Still it is in an initial stage in some places, but especially in the coconut, it is still in a very primitive stage. We are not yet initiated much work on that. That's what.
Yes, sir. Thank you, sir.
Thank
Sir,
you.
and one question, if it's and that is, see, this, as you told it, molecular, this was new initiated in coconut
Yeah.
basis and all.
Yeah.
Is this only for research in laboratories, like for only for developing new varieties, or it will be helpful for farmers also to identify pest before to know the attack of any pest?
No, definitely these techniques are required to develop first at the research level, though it is not for only for research purpose. Okay, whatever we do, this see diagnostic, whatever you can see, even for our COVID or whatever, in the laboratory, initially they have to standardize. Unless they got, then only it will come to public at the usable conditions. That kits when comes, definitely it is very much essential at the farm level to check it. So we should be able to have the kit like antigen kit for COVID or something like that. We should be able to see if the symptoms are there. Farmers should be able to get it tested immediately there itself, whether it is infected with phytoplasma or particular virus or any other disease, so that it can be, that type of things has to come. Even in the high tech and precision agriculture later stages, these type of things are required. But as of now, it looks it is only in the laboratory. That is the problem. Once it comes to standardize in the laboratory, ultimate aim will be to go for the field. Only in certain field crops like potato and other schemes, it has come already into the field level detection kits are there. Even in banana, it is available. So certain crops, it is there for viruses and other things. But in coconut being a hardy, certain issues will be there in the farms, especially, not only in India, whole world, it is the problem. So that type of works, field level kits need to be developed still. It is not for the laboratory work, but it should be to the farmers also, useful for farmers also.
Sir, for a particular crop, there is a different kit is there, sir?
Which one?
For detecting kit, you told now for potato it's already came, banana it's already
potato
detecting
it will
kit
be
is
separate,
came.
will be there.
That's why there's a different crop.
Yeah, different crops, different, we have to standardize. See, potato, which we detect phytoplasma, the same kit we cannot, or Phytophthora in potato, we cannot detect the same kit using in coconut. Okay, so we need to develop separately for coconut. That will not work. It is a different pathogen, though it is Phytophthora itself, the species will be different. So it is difficult to detect using the same issue, as well as the condition of the inhibitors in the coconut, monophenolics, compared to potato and tomato in the annual crops. It is difficult to standardize that detection. We cannot use the same kit for each crop.
Yes, sir.
Okay.
Any other questions?
Yes, sir. Thank you, sir.
Thank you.
Any other questions are there? students? But I think it's getting late and
Yeah, people will be already 8 o'clock today. Okay. They can write to you or our emails. They can send us if they have any queries or want to know more about any particular coconut diseases or pathogen detection or pathogen, this one progress made or symptoms. They can WhatsApp us if they are seeing, and management, if you want to know more, they can also contact us anytime, no problem.
yes, sir.
Okay.
So shall we wind the session now? So shall we wind the session now?
Yeah, I think if there are no
So-
questions,